Hello everyone, my name is Yichen Ding. I'm a new assistant professor in Bioengineering at UT Dallas. My research topic is advanced optical imaging and computational analysis for developmental cardiac mechanics. Before we get started, I would like to invite you to take a look at this video. In this video, we are navigating through the zebrafish heart and investigating the cardiac structures. Besides visualization, you could also measure the distance between two valve leaflets, like this. We entered the heart through the atrium, and now we are walking inside the ventricle. The complicated structures in front of you are trabecular, and their branching pattern helps to maintain cardiac function. I want to draw your attention that this is a virtual reality video, which means all the movements were not predefined, and the user could determine their perspectives by themselves. We recorded this video when one of our collaborators was using our method to investigate the heart. Although this is a prototype, it demonstrates the feasibility that we could integrate biology, imaging, and interactive quantification together in this kind of digital platform. In the future, we could also incorporate fluid dynamics in this platform and integrate experimental results with simulation together. To make this happen, we have three different aims in our lab. First, we need to build our own imaging system, which is a cutting-edge optical microscope. We call it one-sheet fluorescent microscope. Second, to differentiate myocardium from the background noise, we need to train the computer to automatically recognize cardiac pattern. In this case, we developed a machine learning method named SegTransform to enhance the efficiency of image segmentation. Last, we use virtual reality as a platform to build an interactive virtual lab. In this platform, we could digitally test our hypothesis and incorporate simulation into our model. The reason why we would like to develop this kind of methods is we are interested in cardiac injury and regeneration. As you know, heart failure is a global pandemic affecting at least 26 million people all around the world. No matter systolic heart failure or diastolic heart failure, it involves morphological and functional changes. Our human beings do not have the capability to regenerate cardiomyocytes if they were injured. So that's why heart failure happens. However, zebrafish and neonatal mice have the capability to repair the injured heart, even though you cut 1/3 of the ventricle. But in adult mice, they lose this capability again. The underlying mechanism of cardiac repair and regeneration is still unknown, so that's why we would like to provide some new insights from our engineering perspectives. As I mentioned earlier, we built an optical imaging system in the lab. We have 4 different laser lines to illuminate the sample from opposite directions and detect fluorescence perpendicular to the illumination plane. This requires you have strong background and interest in mechanical engineering, optical engineering, and computer science. Once we build the imaging system, we could use it for mouse heart imaging like this video shows. Without mechanical sliding, we could visualize the ventricular cavity inside the heart. We are able to image the whole heart within 1 minute. To make this happen, we also include another method called tissue clearing to render the heart transparent or translucent. So that's why you could see the grids behind the heart. If you are interested in live imaging, we are also able to use this imaging platform for in vivo imaging of the zebrafish heart. In this video, we could capture cardiac contraction and blood flow from the atrium to the ventricle, and we could further analyze the velocity pattern of blood cells inside the heart. The second project in our lab is machine learning-based image segmentation, which is our partners in Los Angeles to develop a method named subspace approximation with augmented kernels, that is SegTransform, to perform image segmentation for our image dataset. The reason why we would like to develop this method is we have very detailed structures in the ventricle, and it's very time-consuming to manually label them. We spend almost 20 hours to label this single 2D slice pixel by pixel. So can you imagine if we have hundreds of thousands of images like this in the heart? That means it's almost impossible for us to prepare enough training data for some popular deep learning methods such as convolutional neural network. Our method is basically based on principal component analysis, and we use it to extract features from raw images and apply random forests to classify all features. This allows us to dramatically reduce the number of training images. I'm not going to include too many details of this method in today's talk, but please contact me if you are interested. The last part of my research work is virtual reality-based interactive quantification. We hope to use virtual reality or augmented reality as a platform for interactive studies. We have already developed a pipeline from our imaging system to virtual reality demos. Once you follow this pipeline, we could convert your animal model to an application on your cell phone. You just need to download and install this application on your cell phone and combine it with a virtual reality headset, such as Google Cardboard. And then you will have an immersive feeling and unlimited perspectives to investigate the example. We further use this contracting heart to demonstrate the feasibility of the interactive environment of volume change in a 4D heart, and it paves the way for us to integrate all our engineering methods with animal models in the future. We will further include our previous method of displacement calculation in this platform to generate a comprehensive and interactive string analysis in the live animal models. To summarize our research work, first, we built our own imaging system named MySheet microscope. And second, we developed a new method named Sec Transform for image segmentation. Finally, we use virtual reality as a platform for interactive visualization and quantification. Even though we use all these methods for cardiac studies, we could use our imaging methods for neuroscience, cancer, and material science as well. Our computational methods are also compatible with CT, MR, and ultrasound. So we believe in future, the integration of imaging and computation with transgenic animal models will provide new insights into developmental cardiac mechanics. Finally, I would like to acknowledge our collaborators and funding agency. Right now, in my lab, we still have some open positions for students. If you are interested, please feel free to contact me. Thank you.